get it done together. All right, let's see. This chapter here is chapter 5 in Gen Bio. Highly recommend, of course, you go home and read your book on that one. I had students who came to me after, let's say, chapter 12 or 13, after they're struggling with the material, telling me, I wish I knew chapter 5 better. Because they realized, had they really studied chapter 5, they would have had a much easier time on chapters 6, 7, 8, 9, 10, 11, 12, the rest of the way. Well, it's a little too late for that. So I tell you guys, there's no wishing. Because when you say, I wish, which means sometimes it will, actually most of the time, it will not come true. It's in your hands. Go home and study it. So then there's no wishing. Just go get it done, please. So we talk about macromolecules, very important chapter. And there are 4 macromolecules. They are carbohydrates. Those are your sugars. Lipids. Those are your fats. Proteins. And nucleic acids, like DNA and RNA. Those are the 4 main types of macromolecules. What does the word macro mean? Large. So notice what we did. We went from the atom to the element to the molecule, the hierarchy of life. Now we're at the point of macromolecules. Again, a link like this means you can go watch a tutorial on it. All right, so make sure you see if you can do that. It helps you. Now, out of the 4 macromolecules, only 3 of them are made from what is called monomers. And the 3 are carbohydrates, proteins, and nucleic acids. Obviously, I did not point to lipids, so no, not lipids. So what is that? Monomers are the subunits of life. So the subunit of a carbohydrate is called a monomer. A subunit of protein is called a monomer. That's the general word for it. The subunit of a nucleic acid is again called a monomer, but they have different names. For example, the monomer of carbohydrate is called a monosaccharide, and you'll see that on the screen real soon. A single sugar, monosaccharide. The monomer of nucleic acids is called a nucleotide, and you'll see that on the screen. The monomer of proteins are your amino acids, the 20 of them. But no monomers for lipids. So they're coming, as you'll see. Again, monomer of carbohydrate is monosaccharide, single sugar, monosaccharide. The monomer of DNA or RNA is called a nucleotide, and you'll see it. The monomer of proteins are the amino acids. Okay, if you have a monomer, you can take a whole bunch of monomers, put them together to make a polymer. Poly means many. So, this is when I start showing you using my markers. Watch the show, please. This is a monomer of a marker. Forget about biology for a second there. This is a monomer of a marker. That means how many? 1. Here's another monomer. All right, now watch me. Watch. I will do this. Now it's a dimer. You see that? Monomer, monomer. Now it's a dimer. What did I do? I linked them together. That's equivalent to a covalent bond. So when we talk about sugar, the monosaccharide plus another monosaccharide, link them together through a covalent bond. Now you have a disaccharide. Are we catching that now? So we're going to speak in general terms first, and then we're going to attack each one of them, the sugar, the lipids, and so forth. So monomer, monomer, bond, dimer. What would this be with 3 of them? Trimer. Let's keep going. What would this be? Not quad. The other 4. Tetramer. Good. What would this be? 5 would be what? Pentamer. What would 6 be? Hexamer. What would 7 be? Don't say like my son. He said sevenamer. Nice try. It's actually heptamer, octamer. Forget it. We're not gonna go there. Watch this. Monomer, dimer, trimer. Sounds good? For me, everything else after that is a polymer. How is that? We don't need to memorize all those things. All right, so monomer, dimer, trimer. Beyond that, it is a polymer. Polymer means many monomers attached to each other. Now, that's a chemical reaction. That is a chemical reaction doing this. Now, watch the show, please. This is where it starts getting fun here. Huh? There's a hydrogen group sitting right here, hydrogen. And there's OH right here, hydroxyl. The way this is right now, it is happy fulfilling the what rule? The octet rule. The way this is right now, it's happy fulfilling the octet rule until an enzyme comes around. An enzyme, which is a protein, it will come around, it will grab onto this guy, grab onto this guy, and do this. Let me have that hydrogen. So once it takes the hydrogen, now this guy is not happy because it's no longer fulfilling the octet rule. And it will do the same thing to this guy. Hey, let me have that OH. H, OH, H, OH, H2O. What did it just produce? Water, H2O. H and OH. You just produced water. So the enzyme will take a hydrogen from here and an OH from here and release it as H2O. Now the problem is this guy says, I'm no longer happy. And this guy says, I'm no longer happy either. Hey, let's share electrons. You see? Now we're happy. When they share electrons, what type of bond is this? What is it? Covalent bond. And then the enzyme says, okay, my job is done, and it leaves. Are we catching that? Okay, let's do it again. The enzyme grabs onto this. What is it removing from here? Speak up, please. Hydrogen. What is it removing from here? OH. What is it releasing? Okay. What bond is being created? A covalent bond. It just keeps doing it. So, in a trimer, how many waters were lost? 2. In a tetramer, how many waters were lost? 3. You see that? And that's how it builds. And that is called dehydration synthesis. Because you're losing water. That's called dehydration. And you're making something, synthesis. So it is referred to as dehydration synthesis. Or dehydration reaction. There's also another term for it, just in case you see that one on the exam. Condensation is another term for that. Condensation, because you're releasing water. Condensation. So, an enzyme is involved. Do you agree? Good. Another enzyme will come in. Watch this one now. Watch. It's going to reverse. Another enzyme will come in, and it will do this. Break. But before it lets go, it's going to take water and give this guy back its hydrogen and this guy back its OH, and then let it go. So you're breaking by water. So let's do it again. Another enzyme will come in and grab onto this guy, the dimer, and break it. But before it lets go, there's water. It takes the water, takes the hydrogen from water, gives it to this guy, takes the OH and gives it to this guy, and lets them go as monomers. So what did you do? You broke it into monomers using water, and that's what this is called, hydrolysis. Breaking or lysing by water. So dehydration synthesis or dehydration reaction is going from monomers to polymers. Hydrolysis is going from polymers to what? To monomers. Yeah.
You said another word for dehydration.
Condensation. So this is the overview. And here's a picture. Here are tutorials on it, please, if you want to see that. So this is similar to what I was doing with my markers up here. Here are 3 markers, and here's the 4th one coming in. Look at the picture, please, so you can have a mental image of it. The hydrogen and the OH, that's what the enzyme is doing. Take the hydrogen, take the OH, release it as H2O. Because you're losing water, it is called dehydration, right? Or condensation. But you're doing it to make something. So it's called dehydration synthesis or reaction. Because now you made this polymer. You went from 3 to 4. Hydrolysis is the reverse of that. The enzyme is going to come in, sit right here, and break that bond. Because covalent bonds are tough, and you need an enzyme to break it. But then it will give back the hydrogen, give back the OH, and release everything. And that is called hydrolysis, breaking by water. Lysis means breaking it. Okay, so that was the general view of how monomers come together to make polymers, and how polymers break apart to make monomers. And that applies to carbohydrates, to proteins, and to nucleic acids, but not lipids, because lipids are not polymers.